Hi, we are Team GenomeCraft, from the Department of Biochemical Engineering, University College London. For the Global Open Genetic Engineering Competition 2024, here we present our project Semi-quantitative Biosensor for naked eye detection of arsenic contamination. Arsenic is a metalloid element typically found in the Earth's crust and groundwater. It is released naturally mainly from volcanoes and hot springs, and artificially as byproducts of smelting. Arsenic is widely used in many different production processes throughout human history, from pesticides, traditional medicines, alcohol, to electronic circuits. Perhaps arsenic is most known for its toxicity. In biological system, arsenic exists as inorganic compounds. Due to its similar configuration to phosphate, the oxyanion arsenate is a competitive inhibitor of many phosphate-dependent enzymes. Arsenic is in fact so toxic that, while uptake is generally unintended, arsenic detoxification systems are found in nearly every organism. For human, arsenic can cause many life-threatening diseases, from nervous system disorders, cardiovascular diseases, to diabetes and even cancers. Unfortunately, hundreds of millions of people worldwide are affected by arsenic contamination, making it a global health issue. While tests for arsenic are available, they often require either laboratory facilities or expensive chemical-based test kits. Such requirements mean detection of arsenic contamination is likely inaccessible at where it is needed the most, such as in rural areas or in household settings. Such issues are therefore our motivation to design a synthetic gene network in E. coli. Here, we transformed E. coli cells with a low arsenic reporter plasmid and a high arsenic reporter plasmid. Upon exposure to samples with different concentrations of arsenic, our engineered E. coli cells produce different colored compounds that are visible to human naked eyes. So, how does it work? First, let's take a look at the plasmid that reports low arsenic level. In the absence of arsenic, the promoter PRs drives the expression of ARSR, which binds to the ARSR binding site, ABS, upstream and inactivates the promoter, forming a negative feedback loop. Upon influx of a small amount of arsenic, arsenic binds to ARSR and prevents it from binding to ABS, therefore activates PRs. This allows expression of the gene cluster VioABCDE, which codes for biosynthesis of the pigment violaceiin that appears blue in ethanol extracts. PRs also drives expression of ARSB, a membrane protein that transport arsenic out of the cell. This system also employs a second plasmid for reporting high arsenic concentration. First, the presence of arsenic activates the first PR's promoter, expressing the recombinase FLIP-E. FLIP-E then inverts the double-strand DNA in between its recognition sites FRTF and FRTR, shutting down its own expression while bringing the second PR's promoter onto the other strand. The high arsenic level means there's still arsenic lingering inside the cell, which activates the second PR's promoter to express the recombinase CRE. Through similar mechanism to FLIP-E, the third PR's promoter is brought onto the other DNA strand. Again, due to high arsenic level, this third PR's promoter drives expression of the chromoprotein E for red, as well as the protein SIL-R. Upon expression, SIL-R binds to the promoter PU of the low arsenic reporter plasmid. This activates PU and drives expression of anti-mRNA of the gene cluster VioABCDE. These anti-mRNAs suppress further violaceiin synthesis, therefore enhance the contrast between the red color of F for red in high arsenic, and the blue color of violaceiin in low arsenic. How are we planning to generate these plasmids in an experimental setting? This slide presents the design of the low arsenic reporter plasmid. The foundation of this plasmid is the PUC 57-amp backbone, selected for its high replication rate and stability. The plasmid includes a PR's promoter and its upstream regulatory site ABS. Downstream of PRs, we inserted the genes for the transcriptional repressor ARSR, the arsenic exporter ARSB, and a gene cluster VioABCDE for violaceiin biosynthesis. To complete the design, a PU promoter is positioned downstream of VioABCDE in the opposite orientation. For construction of the high arsenic reporter plasmid, the backbone PUC57 CAN is chosen. The plasmid includes a PRS promoter upstream of a flip gene, situated between two flipase recognition target sites to enable DNA inversion. A similar system that includes a reverse-directed PRS promoter and the Cre recombinase gene, enclosed by two LOXP sites, has been integrated. Downstream, genes for the FR-red chromoprotein and the CILR regulatory protein are inserted. For both plasmid, construction was done by in silico Gibson assembly. Next, we would like to transform E. coli cells with these assembled plasmids. Firstly, plasmids are introduced into E. coli DH5-alpha cells by heat shock. Cells are then allowed to recover in antibiotic-free media, then plated on LB agar with selection antibiotic. Selected colonies are grown in media with selection antibiotic included to amplify transformed plasmids. To verify correct plasmid construct and successful transformation, plasmids are extracted and sequenced. Additionally, BLAST is also used to assess assembly accuracy and identify mutations. Of course, we need to validate the performance of our system. Since our biosensor responds to the arsenic levels found in water by a color change, we need to establish a clear quantitative relationship between the biosensor's output and arsenic concentration. To do this, we can incubate the biosensor with a series of arsenic solutions of known concentrations. By measuring the intensity of the color change that occurs, we can correlate this with the arsenic levels, forming a calibration curve that allows accurate measurement of arsenic contamination in unknown samples. Following this, we need to assess the sensitivity and the detection limit of the biosensor. By testing gradually decreasing concentrations of arsenic, we can identify the point at which the biosensor no longer shows a noticeable color change. This helps us establish the biosensor's sensitivity and its lower limit of detection, which is crucial for reliable real-life detection. Our biosensor needs to specifically respond to arsenic and not to other common water contaminants. To test for specificity, we expose it to other common water pollutants. This is to ensure accurate real-life arsenic detection and prevent false positives. We also want our biosensor's output to be reproducible. To ensure reliability, we conduct replicate experiments with the same arsenic concentration and analyze the results' variation statistically. In our time course experiment, we focus on the dynamics of the biosensor's output color over time. This helps us determine the optimal time needed for the biosensor to provide reliable and timely arsenic detection in a sample. To bridge the gap between controlled lab conditions and real-world complexities, we expose the biosensor to actual real-world water samples and assess the biosensor's performance. Cross-validation is also important. We compare our biosensor's results with those obtained from established arsenic detection methods, such as atomic absorption spectroscopy or mass spectrometry. These tests provide an external validation of our biosensor's capabilities. Furthermore, we assess the biosensor's response to arsenic under varying temperatures and pH levels. Understanding how these environmental factors affect performance ensures the biosensor's effectiveness and reliability in different real-world conditions. Lastly, long-term storage testing. How does the biosensor perform after being stored for an extended period? This is crucial for determining its shelf life and reliability over time, giving us insights into optimal storage conditions and practical lifespan. To investigate the feasibility of our proposed gene network, we employed mathematical modeling. Our model consists of 14 ordinary differential equations to describe the dynamics of the system's components. Here is an overview of the network in our model, which is a simplified version of our proposed gene network. The model inputs extracellular arsenic level, then outputs the expression of Biosensor and FR-red over time. We used the relative levels of the two colored compounds to determine the final output color expressed by the E. coli cells. Simulation of the model shows that if the initial arsenic level is low, then after a certain time window Biosensor is the dominant colored compound and the ethanol extract would appear blue. On the other hand, if the initial arsenic level is high, then after the same time window, red is the major colored compound, hence the ethanol extract would appear red. We next investigated how the output of our system varies over a range of initial extracellular arsenic level and readout time. Given the same readout time, we found that the ethanol extract color exhibits ultrasensitivity, meaning it switches quickly from blue to red as input arsenic level passes a threshold. Such switch-like behavior supports the ability of our system to provide clear distinction between low from high arsenic level, by reducing the likelihood of encountering intermediate output values. To optimize the system for practical use, it is critical to determine the factors that affect the low-high arsenic threshold, as well as the reliable readout time given a threshold. We therefore examined how these features respond to perturbations of each parameter in the model. Sensitivity analysis shows that both features are most sensitive to changes in arsenic influx and efflux rates. They are also highly sensitive to changes in Rs-B production and degradation rates, which constitute Rs-B expression that drives arsenic efflux. These results suggest that the processes governing arsenic flux could be important optimization handles. How are we planning to optimize our system experimentally? The low-high arsenic threshold is influenced by both arsenic influx and arsenic efflux rate. Since arsenic enters E. coli cells through phosphate transporters, which are likely important for survival, here we instead prioritize optimizing arsenic efflux. Optimization of this feature can be a combination of tuning Rs-B production rate with different transcriptional regulators, and tuning Rs-B degradation rate with different protein tags. Similarly, we can also tune Rs-B production by different transcriptional regulators to optimize reliable readout time. With these proposed experiments and preliminary results, we come to the conclusion of our presentation. In this proposal, we introduced the design for a novel E. coli-based biosensor for arsenic contamination. Our system leverages the expression of violacein and red, providing a cost-effective, accessible, and semi-quantitative method for arsenic testing. We believe our system can provide a wealth of practical values on environmental arsenic monitoring, especially in areas lacking conventional testing resources. For our synthetic gene network, we believe it has very little biosafety risk. We employed E. coli, a risk group 1 organism. For all development processes and potential practical usage, engineered E. coli are not to be released into the environment. Tests will be sterilized with alcohol before disposal. We also limit the activities of recombinases in our system by enhanced degradation. Thank you for your attention. Team GenomeCraft hope you enjoyed this proposal.